What is 16-gene sequencing? 16-gene sequencing is a molecular technique used to identify and classify bacteria and archaea. The 16-ribosomal RNA gene is a component of the bacteria and archaea ribosome, and its sequence varies among different species. By sequencing this gene, researchers can gain insight into the microbiome composition of a sample such as soil, water, or a biological sample. Next, move to the type of bacteria that were used in this research, which is BRUCELLA species. What is BRUCELLA species? BRUCELLA species is a gram-negative, non-motile and facultative intracellular pathogens. This species is from genus BRUCELLA, which is pathogenic to human. BRUCELLA typically affect the reproductive organs, and abortion is often the only sign of disorder in animals. It also known for causing brucellosis, is a zoonotic infection disease that primarily affects animals but can also transmit to humans.
The 16.5 BRUCELLA strains and 17 related strains that could pose a differential diagnostic challenge had their 16S rRNA gene identified. All BRUCELLA 16S rRNA gene sequences were identified to be identical and were clearly different from the 17 related strains, indicating that 16S rRNA gene sequencing is a dependable method for rapidly identifying the genus of BRUCELLA species and distinguishing them from closely related organisms.
The first step is bacteria strain, where 65 BRUCELLA strains representing 6 species from a collection of over 600 were selected to represent temporal, geographic, and source diversity in this study. A panel of 17 related strains likely to present a differential diagnostic challenge was also selected for comparison. All strains were stored at -70 degrees Celsius in defibrinated rabbit blood until testing. The identification of all strains was carried out by standard microbiological procedures. This slide shows Table 1, which presents the designation of 55 BRUCELLA isolates analyzed in this study. Table 2 on this slide presents the designations of strains closely related to BRUCELLA species. The next step is amplification of 16S rRNA genes. Bacteria were grown by planting 1 loop or 1 microliter of salt cells suspension on trap-tithium soil agar with 5% defibrinated sheep blood agar and incubating the bacteria aerobically for 1 to 2 days at 37 degrees Celsius with 5% carbon dioxide. To yield a DNA template, a single colony was suspended in 200 microliters of 10 millimolar trace at pH 8 in a 1.5 ml millipore 0.22 microliters per diameter filter unit heated at 95 degrees Celsius for 30 minutes. The filter unit was centrifuged at 6,000 times cramp for 5 minutes to recover the DNA template filtrate. Each final PCR, which is 100 microliters, contains 5 units of expand DNA polymerase, 2 microliters of DNA template, 10 millimolar trace HCl with pH 8, 50 millimolar KCl, 1.5 millimolar MgCl2, 200 microliters for each DATP, DCTP, DGTP, and DTTP, and 0.4 microliters each of Eubacteria primus F8 and R1492. Reaction mixtures were first incubated for 5 minutes at 95 degrees Celsius, then 35 cycles were performed at 15 seconds at 94 degrees Celsius, 50 seconds at 50 degrees Celsius, and 1 minute 30 seconds at 72 degrees Celsius. Reaction mixtures were then incubated at 72 degrees Celsius for an additional 5 minutes. PCR products were purified with a Quick PCR purification kit. The third step is 16S rRNA gene sequencing, where sequencing primers were chosen from a panel of Eubacteria primers. Among them are F8, R1492, F357, R357, F530, R530, F790, R790, F1068, F1083, F981, and R981. For non-BRUCELLA strains sequenced for comparison, 4 additional Eubacteria primers were used: R1333, R180, R591, and F1127. Sequencing was performed with an applied biosystem peptide terminated cycle sequencing kit per the manufacturer's instructions, except for using 6 microliters of peptide instead of 8 microliters. Sequencing products were purified using centristep spin columns, and these sequencing products were resolved with an ABI model 3100 automated DNA sequencing system.
Next is computer analysis of 60S rRNA gene sequences. First step is to do an evaluation of the 60S rRNA gene sequences of BRUCELLA species. These gene sequencings of the amplified products obtained with PCR, primers F8 and R1492 generated 1,412 base pair long sequences. This allows direct comparison, as the length of these sequences was close to the full length of the gene, which is 1,485 base pair. The sequences generated in this study were then compared with previously published BRUCELLA species sequences by using best fit to determine the level of similarity among them. next step is comparison of 60S rRNA gene sequences of Brucella species to sequences of closely related strains. The U-bacteria primer, F8 and R1492 were also used for the panel of related strains likely to present a differential diagnostic challenge. The 60S rRNA gene sequences from the application products vary in length. For other closely related strains, the sequences were organized into sets by genus and align. A core segment of 1.4 to 1.5 kilobase was selected by trimming sequences at the 5 and 3 ends. The level of similarity of the sequences in a set was then determined by using Jukes-Cantor correction. The sequences within the Agrobacterium set match each other 100%, as did the sequences within the O. anthropi and V. cholera sets. The most divergent sequences within the O. urethralis set match at 99.9%. A representative sequence from each set, as well as the 60S rRNA gene sequences from E. coli, B. hansleyi, A. brumae, and A. felis, were then compared to the Brucella consensus sequence to determine the level of similarity by best fit. In this slide, show the table of similarities of Brucella 60S rRNA consensus gene sequences to 60S rRNA gene sequences of strain related to Brucella species and species important for diagnostic differentiation. The last step is nucleotide sequence accession number. A total of 82 rRNA gene sequences were determined in this study. They have been deposited in gene bank under a number of accessions. Brucella species is under the accession number of AY513506 to AY513568, AY594215, and AY594216.
The 16S rRNA gene sequencing technique offers several advantages in the identification of Brucella isolates. The 16S rRNA gene sequencing can provide specific identification of Brucella isolates due to the sequence of the 16S gene, which is composed of mosaic that include highly conserved variable and hypervariable areas. For some bacterial groups, including Brucella species, the 16S offers genus or species-specific information within specific gene regions, allowing accurate identification. Also, the 16S rRNA gene in bacteria is around 1,500 base pair long, which could provide more information that increases phylogenetic resolution. As I have mentioned earlier, the 16S rRNA gene has varied and conserved regions that change at various rates. The rapidly evolving regions indicate species differences and are helpful for taxonomic classification, while slowly evolving regions allow for the creation of universal primers that amplify genes across different taxa. Alongside the conserved regions, the 16S rRNA gene also has hypervariable regions that indicate sequence variation specific to various bacterial species including Brucella species. These variable areas are useful for species-level identification and for distinguishing Brucella from other closely related bacteria.
The second advantages of 16-rRNA gene sequencing is rapid identification. It has been shown that partial 16-rRNA gene sequencing is suitable for identifying harmful infections. It requires a simple approach that enables short turnaround time, simple handling, and same-day results. Compared to the older techniques, 16-rRNA gene sequencing has become more rapid and accessible because of technological improvement, providing a quick and precise tool for identifying bacteria, including Brucella isolates. Bacteria have highly conserved section called the 16-rRNA gene, which has various areas that can be used to identify species. Researchers can rapidly target and amplify the region of interest by concentrating on this particular gene, which cuts down on the amount of time needed for the study. 16-rRNA gene sequencing has become much faster and accurate thanks to the developments in sequencing technology, that is, next-generation sequencing. This speeds up the identification process by enabling the simultaneous sequencing of several samples. Brucella is among the many bacterial species whose 16-rRNA gene sequences are accessible in large databases. This helps researchers to quickly match the isolate to non-Brucella species and help identifying it by comparing the acquired sequencings with this database. In addition, it also can be processed and analyzed using bioinformatic tools and pipelines after sequencing. While 16-rRNA gene sequencing offers specificity and rapid identification, it is important to note that additional confirmatory tests may be required to validate the results and ensure accurate identification of Brucella isolates.
In conclusion, 16 ribosomal RNA gene sequencing provide valuable insight into the taxonomic composition of microbiome communities, allowing for a comprehensive analysis of bacteria and archaea diversity. It allows researchers to explore not only the structure but also the functional dynamic of microbiome ecosystems. 16 ribosomal RNA gene sequencing provide a broad overview of community composition.